in any case. That's what I forgot to do, but that's okay. So it was Matt Nelson who suggested our program tonight. He says, Michael Kasson, he's got all these studies he does with cicadas and fungi. You have to have him. And I take enthusiastic advice very well. But welcome to Illinois Mycological Association. We're glad you're here.
Well, thank you. I appreciate the introduction and the enthusiasm. And Matt, thank you for suggesting me. Today, we're going to take a voyage and talk about periodical cicadas and their fungi. One, because it's something I've invested a lot of time in over the last 5 years. But the other thing is, come 2024, you're going to have a grand emergence of periodical cicadas in your neck of the woods. And I thought it appropriate and was suggested to me that we kind of link in kind of the work that I've been doing and prepare you for what's to come, not only cicada-wise, but those lovely interactions between obligate pathogens, fungal pathogens, and cicadas. So in addition to being an associate professor of mycology and forest pathology, I'm director of a collection called INVAM that's housed at WVU, and that's the world's largest collection of arbuscular mycorrhizal fungi. For those of you that know anything about mycorrhizae, these are mutualistic fungi that form partnerships with the roots of plants. A lot of ectomycorrhizal fungi are mushroom-forming fungi, which stand in contrast with the arbuscular mycorrhizal fungi, which are, an earlier diverging lineage of fungi that basically sporulate in and on the roots of their host plants. So we have about 900 accessions in our collection. The collection actually is going to be transferred to Kansas here over the next few years. The person who had been running INVAM, Joe Morton, retired, and I took over the collection to act as a kind of a shepherd for the collection as it made its way through the transition. But I don't specialize in arbuscular mycorrhizal fungi. Nevertheless, I understand the importance of the collection, and we're happy to house it at WVU. So just before I get into talking about cicadas and fungus arthropod interactions, I just wanted to say that, at WVU, I do a number of things, and I kind of have 4 pillars to my research program. The first being the historic and emerging canker diseases of trees. And this is really where I got my start, working on beech bark disease. These are members of Neonectria, the genus Neonectria. And I continue to work on those fungi and closely allied fungi to this day here in eastern West Virginia. Of course, I just mentioned arbuscular mycorrhizal fungi, so we do a little work on the interactions with plants. We've recently got into some work between working with hemp and looking at how AMF modulate terpenoids in hemp plants. So that's been kind of interesting. The other area that I've spent a lot of my career on is fungal biocontrol. I did my PhD on killing the invasive tree of heaven with a native Verticillium fungus, and that's gotten a lot of attention, and we continue to work on that. And it's actually become more important lately because of a new pest called spotted lanternfly, which some of you may have heard of. And it's actually, as coincidence would have it, closely related to cicadas. And that's causing some real problems in fruit orchards and vineyards. And its preferred co-evolved host is tree of heaven, or at least one of them. And tree of heaven is in 40 of the 48 continuous states, and provides not only beacons on the landscape for these invasive spotted lanternflies, but serve as bridges across the landscape for these pests to move into new areas where wine production is really important. But the highlight of today will be fungus arthropod interactions. I kind of cut my teeth working on ambrosia beetles, and that's what you're seeing here, an ambrosia beetle in a fungus-filled gallery. These are fungus farming beetles. Did a lot of work on that, continue to work on that to this day. And more recently started to work on obligate parasites, and namely Massospora, which I'm going to talk about today on cicadas. And most recently, we are supported by National Geographic to study the fungal communities of fungus feeding millipedes, a group of 300 million-year-old arthropods that are among the first terrestrialized land animals and may have formed very important, intricate relationships with fungi. And we're investigating that. Maybe that for another day. So here's a snapshot of some of my lab members. We've added a few recently, but I put some green check marks next to some of the people that contributed to this talk in terms of data. Angie Macias is my PhD student who works on the millipedes, as you can see here in the picture, but she did a lot on the phylogenetics of Massospora. My postdoc, Brian Lovett, has been doing a lot on trying to understand the evolution of behavioral modification by these fungi. And just to acknowledge my former PhD student, Greg Boyce, who a lot of this work is kind of built off his chapter of his dissertation, and Matt Berger, a former master's student of mine, who continues to collect weird, rare specimens from me all over the globe and send them to me. So anyway, that's just a quick snapshot of who we are. Now, for those of you that don't know cicadas, it's okay. I'm going to give a little tutorial here. These are different from locusts, although people confuse locusts and cicadas a lot. They talk in the Bible about plagues of locusts. That's not the same thing. Cicadas are different. We're talking about a superfamily in the order Hemiptera. These are true bugs. They feed on sap via piercing mouth parts, and they are close relatives to plant and leaf hoppers. I mentioned just a slide or two ago about the invasive spotted lanternfly, and that's one such example of a plant hopper. That's very cicada-like looking, but here on the top picture, you can see some periodical cicadas on one of my students' hand. These things, periodical cicadas, I'm sure you're very familiar with. And here's an annual cicada below that occurs in your great state. So we'll talk about that in a second. There's about 3,000 species worldwide. Most of them are in the tropics, but we have plenty here to brag about here in the United States. Now, this is really important. There's both annual and periodical cicadas. Now, despite the name, annual cicadas don't complete their life cycle in 1 year, but some members of the population emerge each year. So therefore, we can see them annually. Take, for example, the dog day cicadas, which we hear right now still calling from the treetops. We find them in the hottest part of the summer. There's several genera that are involved with that. But they take 2 to 5 years to develop underground as nymphs. And, there's a lot that happens underground and we can't see it, but we know that they're under there. They're feeding on roots. They're counting sap cycles. And of course, the highlight of today will be periodical cicadas, although we will tie in some annual cicadas too. These are 13 or 17 year cicadas that spend most of their life underground as nymphs, going through a couple of molts, feeding on the roots of plants and counting sap cycles. There's a reason they know when to come up. Cicadas are very good counters, and they basically use the change in root pressure to know when another year has passed. And they have an internal mechanism for tallying those sap cycles, so they know when to come up. And some come up later and some come up earlier. Oftentimes that's plus or minus 4 years. And there's a whole subfield of mathematics almost devoted to the prime number phenomenon associated with cicadas. And it has something to do with predator evasion, but there's been dozens and dozens of papers written about periodical cicadas and prime numbers and how they have kind of found a way to thrive and avoid predation. But they emerge in such high numbers that doesn't even seem to matter. So where do periodical cicadas occur and what broods are present in Illinois? This is a great, this figure up top is a great figure from a paper by Soda in 2013. This was published along with Chris Simon, who's one of the premier, her and Gene Kritsky are two of the premier cicada biologists here in the United States. And they kind of mapped, using genetics, they mapped the kind of different broods of periodical cicadas, where they occur, kind of their gene flow and whatnot. And clearly we could see that there are a number of circles present in Illinois. We're seeing things like 23, we're seeing things like 19, and those are 13-year cicadas. But of course we have some 17-year cicadas as well, including brood 13, which will emerge in 2024. We had a little bit of brood 10, although that was very limited in where it occurred in the state. And that was this year in 2021, the largest brood, although I don't think Illinois got much coverage in terms of brood 10. And maybe someone could clarify a little later on. So you can see that there's a number of broods that are set to emerge in the next few years. But the thing that we want to talk about is, in 2024, we're going to have the overlap of 13-year cicada brood 19 and 17-year cicadas brood 13. So that should be very exciting. And I'll show you here in a second. They don't overlap too much. Now, this statement may come as no surprise to those native to Illinois in the crowd, that Illinois is an interesting state. From a cicada perspective, it's very interesting because it has some challenges for understanding periodical cicada biology. That's because it contains both 13 and 17-year life cycles, and all 7 currently recognized species, including the 3 17-year and the 4 13-year cicadas. And that's 5 separate broods, some of which have disjunct populations. So figuring out who's emerging where, especially when you can have cicadas emerging 4 years early or 4 years late, presents some real challenges. And that's not for most of us to concern ourselves with. People like Chris Simon and Gene Gretzky, and they're doing all that mapping and genetic testing. But we'd like to know who's in our backyard when they show up. And this is kind of a rough map showing their delineation. There's better, newer maps published through UConn's website and with the help of apps like Cicada Safari and community science platforms like iNaturalist. We're able to kind of understand where things are emerging and when they're emerging. And that's real powerful to have that kind of resolved data sets that we can then kind of understand who's where and who's doing what and how populations are changing over time. So I pulled up this is just a screen grab from today of periodical cicadas, and you can look by state, and this is for Illinois. And it clearly shows there's a lot of observations in the northern part of the state. Obviously, most of the cicada action, we're talking in the eastern half of the state. And you can just look here at some of the grabs, some of the pictures, 2020, 2021. So you probably had some brood 10 in there, but the question is that brood 9? Well, no, brood 9 doesn't occur there. So what does that mean that there's all those cicadas in the northern part? Well, those are probably 4-year early brood 13. And that's what you're seeing there, because remember, they're going to emerge in 2024. Sometimes it's tricky to figure out what's going on, particularly when you go to websites like Cicada Maine and said, well, there's no periodical cicadas supposed to be emerging this year, yet you see them. So trying to figure that out. But there's a lot of data available out there, and these types of platforms like iNaturalist and Safari Cicada, they provide an exceptional amount of data for even the average, naturalist to really make sense of what they're seeing. Now, I'd be remiss if I didn't mention that there's some other annual cicadas present in Illinois as well, in addition to the periodical cicadas. And of course, a number of these are some of the screamers you hear in late summer, the neotibus and whatnot, some of these dog day cicadas. But there's some other cicadas as well, and quite diverse. I'm going to talk a little bit today about one of these, Oconagana ramosa. That's one of the annual cicadas that we studied our fungus on. So it's not just periodical cicadas that have a obligate fungal parasite, but these annual cicadas get them as well. And we haven't found them on all the annual cicadas, but there's quite a number of annual cicadas that do get them. And Oconagana ramosa is one of those species. So this is not all the diversity of cicadas present in Illinois. I'd encourage you to check out Cicada Mania, which is a website, all things cicada, and you can look up all the cicadas that are present in any state, and it's a real nice resource. But I really, I'm here to tell you a story that started back in 2016, and really it changed my research program significantly. Oftentimes we're caught in the middle of something, and it's just like an opportunity arises, and we can't pass it up. And really that's what happened with cicadas and Massospora starting in 2016. I started at WVU in 2014. And although I was studying fungus insect interactions, particularly I was studying ambrosia beetles and their fungal mutualists. I didn't really do much other work on insect fungal interactions. But these cicadas started emerging, or at least they were getting ready to emerge. My students came in April of 2016 and said, hey, the cicadas are getting ready to emerge. And two of my students were real cicada enthusiasts. They had t-shirts with cicadas on them. They were ready, but I wasn't ready because I didn't really, this was outside my, I hadn't heard of this. I hadn't looked at it. But certainly when they started to come out in mass numbers, you can't avoid them. They're just everywhere. And if there's one thing we know about disease, is that whether we're talking about COVID-19 or obligate, early diverging fungal parasites of insects, with increased population densities comes, epidemics. And when cicadas emerge in the millions of cicadas per hectare or millions per acre, you're going to see a number of disease epidemics emerge, or epizootics. And that's exactly what we were expecting and what we saw. Not only are there wing deformities based on temperature and other bird strikes and things like that, but it was quick to see a number of the fungal pathogens emerge. So I just want to remind everybody before I get into all this that cicadas have been known for a long time to have fungal parasites. In addition to Massospora, we get things like Ophiocordyceps, which is this mohawk nymph here on the upper left. We get Buvaria, we get Metarhizium, Eiseria. These are all fungal pathogens of different stages. Some are nymph specific, some are adult specific. These two down here are periodical cicadas. This is not a periodical cicada, but sometimes it's hard when you just have a nymph to know what it is, but we know because of the literature. Now, there's other cicadas like these dog day cicadas I was mentioning that have kind of a white underbelly and look like they have fungus all over them, but that's just how they look. And we get a lot of calls about these. We get a lot of people saying we found a fungus infected cicada. And it just turns out it was one of these cicadas that died on a pavement, and they see it in the white underbelly and they think, well, it's fungus. I understand why people would assume that because it's kind of chalky looking, but that's just how they naturally look. So there are things that look like fungus that are not. But my students were like, listen, there's this obligate pathogen of cicadas called Massospora. And Angie, the student pictured here on the right, was an undergrad at Cornell. And she worked in the fungal herbarium up there with Kathy Hodge. And she had photographed the type specimens for several Massospora species. And she said, and she had written a little article for the Cornell mushroom blog on Massospora back then, and coined a phrase that became a darling of the media, flying salt shakers of death. And we'll talk about how Massospora turns cicadas into that. But she said, listen, there's this really cool understudied parasite of cicadas, fungal parasite, and it makes their butts fall off and it doesn't seem to kill them. And I don't think many people have studied it. I think our lab should study it. And Matt Berger, who was also a student, grew up in Cincinnati, spent his life just studying and loving periodical cicadas. He was the one with several shirts. And he said, look, I would love to be involved with this. I'll collect as many as we need. This is something I'm really passionate about. And, eventually they brought me this first specimen and said, here it is. And once I saw it, I recognized that it was really cool because the cicada was still alive. And even though it had a fungal plug popping out of its posterior end of its abdomen, and you can see the abdomen popping open here and this chalky white fungal plug emerging. And the abdominal sclerites are sloughing off in rings. And the cicada can continue to fly, continue to mate. So that was really interesting. So I said, okay, we don't have a lot of time, but let's collect as many as we can. I gave, we had collection vials. Let's put them in the freezer and then we'll figure out what to do next. If there's one lesson I often tell my students and tell people when I give talks like this is that you really have to embrace opportunities to sample things like this, because you can always say, oh, I'll go back, I'll get another time, but that moment will pass. And, I don't go anywhere without collection tubes because you never know when an opportunity to collect something strange might occur or something rare. So we were lucky that this was happening right outside on our campus. So we can literally walk 100 yards and collect dozens of infected cicadas. So what do we know about this fungus Massospora that I want to talk about today? Well, it's a member of the Entomopterales. Some of you may know that that order of fungus because it also is Entomoptera musci, the fly summit disease pathogen is that. Also, there's a biocontrol agent of gypsy moth that is also a member of the Entomopterales. And this is in the Zoopagomycota. This all used to be zygomycetes, but is no longer. So we know of at least 13 obligate sexually transmissible pathogens in this group that infect some 21 cicada species worldwide. And my student, Angie, summarized this in a paper we published last year. You can see these various Massospora species. Now, we highlighted the ones that we focused on for our study, and we'll get into that here in a second. But you can see, a number of these occur in South America. Some have been from Australia, but mostly, the Western Hemisphere, although there are some reports from the Eastern Hemisphere. This was formally established by Peck in 1879. It was described earlier. The first recorded publication is about 1861, but it was formally established by Peck in 1879 with a periodical cicada from New York. Despite its description in the late 1800s, it remained, like a lot of fungi, a mycological oddity for 100 years. Now, it's easy to understand why. Let's think about the insect. It spends most of its life, 13 or 17 years, underground as a nymph. Emerges in a spectacular fashion for maybe 4 to 6 weeks. And only maybe 1 to 5% of the population, on average, is infected. So you have to be in the right place at the right time, and you have to wait long periods of time in between emergence. Now, luckily, we live in a time and an age where it's easy to drive from one location to the next. It's a lot harder in the late 1800s to drive around chasing the different broods because it wasn't well understood what the different broods were, or where to go and sample. So it's easy for us as modern day scientists to say, okay, last year we traveled to southern West Virginia. The year before that, we traveled to, Southwest Pennsylvania. And before that, Syracuse, New York. And before that, we followed the different broods because being in West Virginia, we're centrally located and we can collect fungus infected cicadas each year. We have to travel a little bit. But if you were a scientist back then in one place, it would be really hard to make progress on something you can't rear in the lab and that only shows up once every 20 years. So from 2016 to 2019, we acquired a lot of infected specimens. Here's just the 3 Magicicada species, both male and female, males on the bottom. You can see the tymbals behind the back legs. These are the sound producing organs that only males have. And you could see the Massospora infections on each of these just to see kind of how they manifest. So this is male-female pairs for each of the 3 Magicicada species that are part of brood 5 and are part of many of the broods. Some broods have fewer than 3, but a lot of broods have all 3 of these species. But we also acquired a number of infected Massospora species or Massospora infected specimens from various collaborators. I told you my former student, Matt Berger, would go hiking and collect things. So we were able to basically have people rummage through their freezers and, insect drawers. So we had dried specimens, we had alcohol stored specimens, we had fresh collected specimens from out west, and -80 specimens from Chile. And, so all these things, and we were able to pull them all together and answer some really interesting questions. And we can see the fungal spores here. We see the canidia on top, and we see the resting spores on the bottom. And we'll talk a little bit about the life cycle of the fungus here in a second. This is 5 different species of Massospora that we were able to acquire and kind of study.
Matt,
Yeah.
Pardon me, we have a question in the chat, and it's actually from Sarah's 8-year-old son, but maybe others in the audience have the same question.
Yeah.
Sarah's son wants to know how the fungus makes the butt fall off of the cicada, please.
Well, I will answer that, but I'll briefly answer it here, and then I'll show the disease cycle. The fungus gets inside the nymph, so it actually infects the nymph when it's underground. The nymph comes up, it molts into an adult. It continues to devour the inside of the abdomen and building up pressure to the point where it starts to break apart the exoskeleton in the back. And the way that the exoskeleton is assembled, it's kind of in these rings. So it makes the backmost rings fall off first, revealing this fungal plug. So it's a combination of using up all the real estate and pressure from the expanding fungal plug that basically it consumes that part of the inside of the cicada, but basically breaks off the outside of the cicada, if that makes sense. So good question, thank you. Of course, a lot of the papers from the 1800s, you're not going to find, well, this is what the DNA analysis told us, of course, because really we didn't start sequencing things till the 1990s, and we didn't really, use ITS widely until much later than that. We're talking like early 2000s and stuff like that. So, if you're going to try to link modern studies with historic studies, you have to go back to the classic morphology. And I'm a strong believer in coupling morphological studies with molecular phylogenetics. I think it leads to more robust science. So we were able to do things like look at the number of nuclei per spore and look at the epispore and for roughness and measure the length and the width of the spores to compare them. And say, okay, these are allegedly 5 distinct species. Do we see differences? And in some cases we did. Like I have circled there, the width showed that for 5 different species, we had 5, based on statistical tests, they were significantly different. But the length, not so much, and the resting spore diameter, not so much. So sometimes things can be morphologically cryptic. That is, we can't tell them apart morphologically, but they still are different. Now, we were really excited because we were able to generate the first DNA sequences for this fungus. There was one deposit in GenBank, but it turned out to be an incorrect deposit. It was an Entomophthora that was deposited. So we were able to deposit these and create a phylogenetic tree. That is a family tree based on DNA sequences so that we can determine whether or not these species boundaries held up. That, in these historic publications said, okay, these are 5 different species. Well, as it turned out, that it resolved into a single group. So these are all closely related. They're all kind of sisters and cousins. But what was really interesting is that 2 of these, Massospora-cicada from these wingbanger cicadas out in California and Colorado, and Massospora-levispora, which came from the Okanagana ramosa, that one annual cicada I pointed out that's also occurs in Illinois, formed basically a clonal lineage. And basically what this molecular data is telling us that these 2 things, although they were called 2 different species, are the same fungus. So that was really interesting to find. And we'll get back to why that's even more interesting here in a second. But, the rest of the data just kind of supported the morphology that, there were distinct species and that all Massospora species were closely related. But we recognize that this Massospora-cicada and Levispora were once called 2 species, are now 1. So that happens a lot in taxonomy. So it's not a big deal, but it was pretty interesting nonetheless. One of the reasons that people really like Massospora, and we like talking about it, is that it modifies the behavior of its host. And you may be familiar with summit disease because it's one that's gotten a lot of attention. Rightfully so, it's really interesting. You've probably heard of the zombie ants. This is the phenomenon where an infected ant, an ant infected with Ophiocordyceps, will climb to a high perch and lock down its mandibles onto a blade of grass or a leaf or a twig, fix its abdomen, and then a fungus will emerge from the cadaver post-mortem, even out of the head, and grow up and sporulate, and those spores will rain down on unsuspecting ants below. And this is a real interesting phenomenon. And this is a type of manipulation done by fungi to increase or maximize spore dispersal. We call that summit disease. The infected arthropod always goes up. It always adheres or affixes to a substrate, and then there's post-mortem sporulation. That is, the fungus produces spores outside the infected host once it's died. That's really in contrast to what we're seeing with Massospora, the cicada fungus, where we have active host transmission. Now, I said we made the kind of funny observation that the butt fell off, but the cicada continues to fly around as if everything's normal, as if its butt didn't fall off. Now, up to a third of its body could be consumed by the fungus, and yet it seems to have an unusual pep in its step. If one of us were to lose a limb, we probably would feel lethargic and want to remain stationary, but that's not the case for cicadas that are infected with Massospora. So the conidial dispersal happens prior to host death, and it's also seen in another fungus called Strongwellsia and Entomoptera. Now, what you're looking over here at the right is kind of like a subway map for behavioral modification. And if we follow Massospora, for example, we could ask the question, does it host behavior facilitate transmission? Yes. Are the spores discharged by the fungus? In Massospora's case, it's not. They're not actively discharged. They fall off. That's why we call them kind of flying salt shakers of death. They pour out of the abdomen and loosely fall off as the cicada flies around and engages in normal mating behaviors. What's the host status during transmission? It's alive. We call that active host transmission. And some cause summit disease, and some have no manipulative transmission. But that's not the only thing that happens with infected cicadas. Active host transmission is a phenomenon we see in several genera, Strongwellsia and Entomoptera, but there's something unique about Massospora. We see what's called hypersexual behaviors in infected cicadas. So in addition to attempting to mate with females, an infected male, for example, will mimic females to attract mating attempts by other males, thus doubling the number of cicadas that an infected cicada can come in contact with. This is present only in cicadas with conidial infections, not resting spore infections. And this hypersexual behavior is known from Magicicada, Platypedia, Oconagana, and Diceroprocta. And that's why we really tried to secure samples of those, and we did get samples of all those for one of our studies. And for the study I'm going to highlight next, we had 3 of these species. So just to kind of show you what's happening, to follow up on that question earlier about, when's infection occurring, you have these nymphs that fall down at the end of the summer off the branches. They go down, they start feeding on the roots, and that's where they're going to remain for 17 or 13 years. And they'll move around. If roots die, they can move to some adjacent roots. But if whole forests are cut down, the nymphs will die underground with the dead roots. So you can see where land clearing in between successive cicada generations could really impact the survival of cicadas in those local environments. But over time, those nymphs start to come up top, getting ready to emerge in the months leading up to year 17 or year 13. They create a vertical emergence chamber where they sit below the soil, like below the duff layer, and they wait for the temperature to reach a perfect 64 degrees, at which they emerge synchronously in the billions. So it's during that time we think they encounter Massospora, and we think the spores are actually interacting with a hormone cue in the cicadas, because otherwise they might just try to germinate, infect anything that passed by. But we think that they've co-evolved to recognize the molting hormone of the cicada, saying, hey, the cicada's coming up and it's ready to emerge. Now's our time to get in. So what happens is the nymph comes above ground, it is infected, it molts into an adult, but it maintains the infection. And then over time, the abdominal sclerites start to slough off, revealing this fungal plug. Mating attempts occur, although not successful, because one of the cicadas no longer have genitalia, but instead have a fungal plug. And then that transmission occurs through a mating attempt, and the cycle kind of continues. Now, the cicadas that receive the spores from the mating attempt actually develop resting spore infection, and that eventually makes its way back into the soil. But that's generally the life cycle. There's some components where we don't actually know if certain things happen, so we put a question mark, because We don't know, for example, if a cicada that makes contact with an infected cicada, if it gets its own conidial infection, and that can happen twice before it goes to resting spores. We don't know if females could give their nymphs Massospora infections before they actually go down underground. So that's a really interesting question too. So one of the interesting things about this is that the hypersexual behavior seems to be confined at the conidial state and absent from the later stage. And we did kind of confirm this through chemical analysis, and I'll talk about here in a second. So because a lot of work has been done on behavioral modification in animals and fungal parasites, we thought to ask whether or not small molecules, that is fungal secondary metabolites, might be responsible for these aberrant behaviors we saw in the cicadas, because really it's only occurring in a specific spore stage of the cicada, and it's present in more than one cicada species, and it's always kind of the same. So really we're asking, can fungal metabolites be responsible for this? And are those molecules conserved across Massospora species? So what we did is we applied basically something called global metabolomics, and this is using liquid chromatography mass spectrometry. We don't have to get into all the details of that, but this is a highly sensitive instrument. This is analytical chemistry, essentially, where you can basically pulverize a sample, and that we basically took the fungal plugs off the back of the cicadas, and we put them in, we put them in methanol, and then spun it down, and then ran it through this LCMS, and asked what kind of compounds are present in those plugs. And we were able to do that for both Massospora-cicada plugs, and we have these Massospora levospora plugs as well. And we also had some resting spores in Massospora-cicada. And when you look at it visually, you can kind of look at these total ion chromatograms and see kind of different signatures. This is a Massospora from Periodical cicadas. This is a Massospora from annual cicadas, and you can see they look very similar. And then compared to the abdomen of just a healthy cicada, which looks very different in the middle. So you can see that these kind of fingerprints are more closely related than this. So each sample produced unique signatures with two Massospora species looking more identical to each other than either one of them looked to the healthy cicada. And of course, with these kind of global studies where you're asking who the heck's in there, you're going to get a lot of stuff. And we found 1,176 differentially detected small molecules. And we can get that to spit out in an Excel spreadsheet. You just scroll down and you kind of look around and say, okay, what did it find? And some of the things are well-known compounds, some of them are not, some of them are just chemical formulas, some of them are just masses. But two alkaloids stood out to us right from the start. So this is a couple years ago, and I'm sitting at home and my phone rings on a winter like really early in the morning, and it's my grad student, and it's Greg Boyce, and he's the one who kind of helped. He and I made this discovery, and of course I'm reaching for my phone because it's really early in the morning. I'm trying to figure out what's going on. And it's Greg in the line. He says, Matt, I got to tell you something. I'm like, Greg, what time is it? He's like 2 a.m. or something. I'm like, Greg, what's going on? It's 2 a.m. Like, what are you calling me about? And he said, those Massospora plugs that you gave me from the annual cicadas out west, the wingbanger cicadas that had the Massospora platypedia. I said, yeah. He said, well, you're not going to believe this. They're loaded with psilocybin. And I'm like, what? And he said, you know what psilocybin is, right? And I said, of course, that's the psychoactive compound that's found in magic mushrooms. And I said, like, are you sure? And he said, no, I'm sure. It's in every sample. It's in every sample. And we published this back in 2019. He said, but we have to do some other things to confirm it. And I said, okay, like we talked for a couple minutes and then hung up. But, of course, my mind was really, and after that, really excited that, we had basically detected this high amount of psilocybin from the Massospora levospora, but not from the cicadina or the control. And it was 17-fold higher compared to those other two. So the next thing you basically do is you try to compare it to a chemical standard. And you look at its fingerprint, the way that you compare, fingerprint at a crime scene with a fingerprint database. And you ask, okay, if we apply a voltage to this mass and we fragment it, it creates this kind of unique fingerprint. And then we compare it to this analytical standard you can buy. And sure enough, it matched 8 fragments. And this is like complete confirmation that the compound is there. And it wasn't present in the healthy cicada abdomen. Now, one hiccup we hit along the way is I went to go order this compound, the Psilocybin analytical standard. And of course, as I put it in my shopping cart at like, one of these online scientific stores, this warning stop sign popped up and it said, please enter your DEA research number. I was like, oh my God, like this is regulated by the DEA. We can't like study this. Like, and my student assured me, he's like, don't worry about it. We'll just get a DEA exempt analytical standard. It's really common. You can get ones that are like, can't be abused. So you can just get one and you don't need a permit. So we were able to do that. And, that was no problem. So of course, my next question, and maybe your next question is, okay, it's great you found this in some cicadas out west, but like, we're getting ready for periodical cicadas. Let's hear about periodical cicadas. So of course, we went right over to the periodical data set and said, do we see the same compound in periodical cicadas? It wasn't there. And I was like, oh man, what are we gonna do? So we're digging around and then we're sitting in my office and Greg's at the other desk and he said, wait a minute, I think I found something. And he said, have you heard of cathinone? And I said, no, I haven't. So quick, did what most scientists do, got on Wikipedia and pulled up cathinone. So if those of you that don't know cathinone, this is a naturally occurring amphetamine or stimulant that occurs in a plant that's native to the Horn of Africa called cat plant. And we detected this compound in Massospora-cicada conidial plugs. And they chew this in parts of the Middle East, including places like Yemen. It's used as a stimulant to engage in conversation. And it was definitely like really highly upregulated in Massospora-cicada. We did find a little bit in Massospora-platypedia that was later determined to be not true. We basically followed up with an experiment to figure that out. And there's basically something called isobaric compounds, and I won't get into that, but that wasn't legitimate. But what you can see here is that it was much higher in the conidia than it was, say, the resting spores. And what that's telling us is that if this is a candidate compound for behavioral modification, it's only happening in the conidial state. And that's actually where we're seeing the behavioral modification. So that was really interesting. So of course, we did the same thing. We found a DEA exempt standard. We fragmented and confirmed that at least 3 of the fragments matched. And really all you need is 2 or 3 fragments to confirm that, as long as they're within this kind of acceptable error limit. And we didn't find it, most importantly, we didn't find it in the healthy cicada abdomens.
Matt, we have a question on the DEA exemption on the standards. And the question is it similar to the research alcohol tax exemption where they just add poison so people don't consume it?
Yes. Yeah, it's exactly what they do. They add like acetonitrile or some kind of compound that if you consumed it, you would die upon you consuming it. So yes, it's exactly like that. As kind of since we did kind of stop to talk about the DA thing, I will mention the story because it's actually one of the funniest stories like of the whole project, and one that I talked to several journalists about when we got a lot of coverage on this, is that after these discoveries, I realized that I was sitting on a bunch of specimens with amphetamines and psilocybin in the lab. And I'm just like, I'm a law-abiding citizen. I'm kind of a straight arrow. And I was just like, oh no, the DEA SWAT team's gonna come in here. They're gonna tase me, steal all our samples. Like, I was really nervous because you're not supposed to research this stuff if you don't have a DEA permit. So I sent the most awkward email in my life, basically self-reporting to the DEA. And I get this email back and it's just like, this is a very strange situation. Let me consult with the chemist, the main chemist for the DEA. And it was ultimately determined that, we couldn't have expected this discovery. We weren't trying to concentrate it or enhance it or sell it or distribute it. So we were in the clear, but we felt like we needed to self-report if we're going to publish it. But it was certainly like a weird thing to have to like interact with the DEA, you know, studying cicadas and fungi. And then, you know, because we had these standards, we were able to create standard curves and quantify the amount of psilocybin and cathinone and these things. And it showed that, you know, most of the plugs had, you know, and this comes back to, if you remember, I said when we did the molecular studies, we basically saw that two of the cicadas fungi were identical. And as it turns out, both of them had psilocybin. So it was kind of an independent confirmation that it was the same fungus because they both contain the same chemical compounds too. So that was really cool. Now one contained psilocin and one contained psilocybin, and you know, the one's a breakdown product of the other, but they're the same compound. So that was pretty interesting. And the levels were more or less, you know, between 0 and 15 or 0 to 20 nanograms per fungal plug. Now, a question that's often asked is, how many would I have to eat to have a, you know, good night? Well, that was just one of 1,176 compounds we found differentially expressed or detected in these plugs. And some of the things we found were toxins. So, I mean, you know, we know what's in magic mushrooms. They're consumed recreationally and now medicinally. But we don't know all the compounds that are present in these fungal plugs of cicadas. So I would definitely not go out of my way to try to consume one or more than one. That said, if your dog swallows one, I think your dog's going to be okay too. But these compounds were absent in healthy cicadas. So cathinone, a similar story, but actually we found a number of negative samples. And if you understand the breakdown of metabolites, something like psilocybin has a longer shelf life than an amphetamine. And all you need to do is think about how these drugs are used recreationally. Amphetamines tend to be short-lived. We know that like recreational drugs like bath salts are a type of synthetic cathinone. We also know that drugs that are used for ADD and ADHD are a synthetic cathinone too. So these drugs have utility from a pharmaceutical standpoint, but bath salts have been implicated in some really kind of strange behaviors. But getting back to the point that a number of these fungal plugs were absent of cathinone, but probably because of its short half-life, it just had deteriorated. So, you know, what's great is that in the literature, if you go back to the 1960s and 70s, there was a lot of studies where they decided, you know what, let's just drug a bunch of insects with everything and see what happens. So there's studies where they give LSD to spiders, and they give amphetamines to ants, and they give you know, peyote to spiders, and all kinds of drugs to see how it affects web building, see how it affects aggression. And what some of these studies showed is that the doses that we found in cicada plugs, like for example the cathinone, was enough to cause aggression in ants, and it also altered the feeding behavior in adult blowflies. You know, let's think about like a number of these kind of drugs not only have a targeted impact where they might like increase attention and things like that, they also tend to suppress appetite because you're focusing on the one thing you're supposed to do. And if you're a fungus trying to manipulate a cicada, you don't want it to eat as much. You just want it to focus on mating and focus on spore dispersal. And that's what these cicadas are. They're extended phenotypes of fungus. And we know, for example, that psilocybin ingestion has a less observable impact. Spider drug webs took human appropriate dosages for it to be messed up. But there may be some tie-in with some of these cicadas out west that rely on kind of wing tapping. And we know that temporal perception is messed up by things like psilocybin. So we're still trying to understand all this and really figure it out. But the problem is the next steps require direct dosage or dosing of cicadas, and that does require DEA oversight. So we haven't taken that next leap because the permits involved are extensive and require a lot of time to get approval. So, but really the compounds present in these like independently sampled populations provide some evidence of a chemically induced behavior. The neurogenic activities of these compounds do explain some of the behavior, some of the prolonged wakefulness, and some of the the hypersexual behaviors that we see in these cicadas. And maybe both may act to suppress appetite. But, they can't explain all these things. And we know that, it's not always so simple as it's one thing. There's interactions with hormones and things like that. We know that the physical act of castration, for a male cicada to have its genitalia taken off by this fungus is going to affect hormonal cascades, and that may be interacting with the fungus somehow. So there's a lot to be learned still. And with regard to what's happening underground, there's a lot we don't know that's happening underground. It's hard to observe the life cycle under there, and we can't rear these in the lab. So there's a lot of questions we hope to answer each year, and we only have a short 4 to 6 week window to ask them and answer them. So we're continuing, we're trying to sequence the genome of this fungus. We sequenced the genome, but it was very low coverage. Turns out that these genomes are 10 to 20 times bigger than most fungal genomes. So it takes a lot more to sequence them. And we're doing some follow-up, confirming the compounds and the plugs, and doing some other things, continuing always to look for more Massospora specimens. Now, some of you may have heard of some mysterious disease that blinding and killing hundreds of birds in the DC area, the mid-Atlantic, that coincided with the cicada emergence. These were mostly fledglings, but a diverse number of birds. And although it's probably not linked to cicadas or Massospora, we are considering that it may be involved in some of these bird deaths. So it's there's a lot of collaborative work being done now to see if Massospora might be implicated in some of these bird deaths, or if cicadas, even in the absence of Massospora, are involved somehow. We recently acquired some cicadas from the Florida Keys and from California, which may represent new Massospora species. So we're excited to get some sequence data and some morphological data and do that. At the end of the day, maybe someone might leave this talk and say, okay, well, the fungus is cool, cicadas are cool, but like, how can I get involved? Well, if you look at the map of Illinois and you look at the Massospora data for iNaturalist, there's one observation. So I'm challenging everyone on the call that uses iNaturalist to make sure when the next brood emerges that you're uploading your pictures and your observations of Massospora. You can also do that on Safari Cicada and elsewhere. You can also send us samples, but at minimum, I think making some observations and uploading them to iNaturalist is a really good start for how you can be involved and be part of the community. But we have very little sequence data from Massospora from 13-year cicadas. We have confirmed that the Massospora that infects 17-year cicadas is the same one in 13-year cicadas. In fact, I showed you this tree because GK2 is a 13-year cicada. I think this is brood 23. So it's clearly part of the same group. But some of the data seems to suggest that there's kind of sequence divergence between the 17 and 13-year, but we just don't have enough isolates to resolve that question. So I would love to get some 13-year cicada Massospora infections from this next Illinois emergence here in just a few years. So keep us in your thoughts. So with that kind of wraps up the story of Massospora and periodical cicadas and behavioral modification. I hope this is something that you found interesting, and I am more than happy to answer any questions you may have on cicadas, fungus insect interactions, Massospora, you name it. It's kind of an ask me anything fungus related. Thank you.
Thank you very much for a fascinating presentation. I'm sure we'll have quite a few questions in the chat shortly. But I thought maybe I could start with Matt Nelson. Sorry to put you on the spot, Matt, but you've been dropping a variety of links throughout the program in the chat. Maybe you want to talk about some of those and ask Dr. Matt Kasson some questions in particular.
Yeah, they were They were first off, that's really cool talk, Matt. Thanks so much for your time. I know you're on East Coast time too, I'm guessing, so it's especially late for you. So
it's fine. Thank you.
But yeah, the links I was just putting in were links to a couple of the papers you were mentioning, the Plant Pathogens one and a more recent one. I guess one question I had was, have you tried sequencing other species of cicadas and looking to see if Massospora is there in like a latent state at all? Just curious, like, is
We
it
haven't
something
yet,
where everyone kind of has it, but it's just a really...
Well, one of the biggest revelations in cicada fungal interactions in the last few years is came out of like John McCutcheon's lab. And they basically, you know, cicadas, like a lot of and a lot of Hemiptera, have bacterial symbionts in their guts. They have a special organ called a bacteriome because they feed on sap, which is nutrient poor, and they need to supplement with amino acids that are provided by these bacterial symbionts. Well, in a number of cicadas, the bacterial symbionts have been displaced by an Ophiocordyceps. Now, if you know Ophiocordyceps, that's a pathogen typically, but it's a domesticated Ophiocordyceps. So they've basically tamed an insect pathogen to provide basically nutrition in the absence of bacterial partners. It's possible that Massospora may play a similar role in some cicadas, and then there's a trigger that basically it goes from a latent or a facultative. No, maybe it's a dominant mutualist and a facultative pathogen, and then in that adult stage it emerges. So that's a really good question, and one that we collected some eggs and some emerging nymphs to ask that question. In areas where we found a higher incidence of Massospora infection, we want to see if we can detect Massospora in surface sterilized nymphs that were on their way down. So it's that's great thought.
I currently don't see any other questions in the chat. I guess what comes from doing a very thorough presentation there.
Or long presentation. Maybe.
No. It was just the right length. It was really fascinating. Thank you. I do see that someone here has a question. Weston, can you add the question into the chat window, please?
Or if you feel comfortable, you can ask it out loud.
Yeah, I know you explained it a little bit in your talk, but I was wondering if you could just explain a little bit more about how after they mate, you said like twice, and then the plug falls off and then they die, and then that's what transfers the fungus into the ground. Or could you explain that a little bit more in depth?
Yeah, so the infected nymph that comes up from the ground molts into an adult, and it's an infected adult. Eventually its abdomen bursts open and the fungal plug is revealed. That infected adult will then make contact, a mating attempt with a healthy individual, and they'll back into each other because when they mate, they kind of like back into each other. And that abdomen or fungal plug contact will then lead to a secondary infection. And that newly infected individual will develop not a conidial infection, but a resting spore infection. And eventually that cicada will fall to the ground and those spores will integrate back into the soil. And it's not, it doesn't happen just like that. There's like a week before the fungal plug emerges. And then, the infected individual will attempt to mate with a lot of different cicadas and spread it, it's sexually transmissible. So it's a fungal STD, and they're just widespread spreading this around. And because of the behavioral manipulation, not only are infected males making contact with healthy females, they're also pretending to be females to get males to come in, and they're making male to male contact and spreading the spores that way. One of the grotesque things that happens during fungal infection is females' abdomens don't fall off that easy because they have a lot of muscle tissue associated with an ovipositor. Ovipositor is a sword-like egg-laying organ on the back where they're able to penetrate bark tissue and lay eggs, and they have a lot of muscles associated with that. Well, because that muscle is there, the abdominal sclerites can't come off. And oftentimes, if an infected female mates with a healthy male, when they're done mating, the male will break off her genitalia with him, and he'll fly away with the backside of her body. So it's quite grotesque. But this is the life of fungus infected cicadas. It's wild.
All righty then. Dr. Greg Mueller has a question in the chat. How did the psilocybin get in? Was it through horizontal gene transfer?
Yeah, so that was one hypothesis. The problem, we haven't been able to fully test it because the genomes are so big, we need to get a more assembled genome. And we have no evidence that there's gene clusters like there are in the psilocybin producing magic mushrooms. Now, not all the enzymes needed for, or the genes needed for psilocybin production based on what we know psilocybin production takes in magic mushrooms are present. So it could be an independent evolution of psilocybin production. And we'll need a couple more pieces to confirm that. If that's the case, it'll be a real big story. But yeah, horizontal gene transfer was kind of our first idea. But we'll see what happens there.
So Matt, I was wondering, is psilocybin known outside of this now and magic mushrooms? I just am not aware of other places it occurs, but I haven't kept up with the list.
No, there's really, those are the two instances. There are some genes involved in psilocybin production, but not all of them. There's a termite fungus, and that's been published somewhat recently. Jason Slott's group out at Ohio State. But more or less, it's Massospora and the magic mushroom. So that's why it's something we want to really nail down because it's interesting. I see a question from Matt here. What's the level of selectivity like? Does each Massospora only infect one cicada species? So that's really interesting because we showed that, for example, the Massospora that infects Platypedia putnami in California, New Mexico, Colorado, Utah, is the same Massospora that infects Okanagana species in Michigan. And they're actually sister species or sister genera. We just got a Massospora diceroprocta from a Florida Keys cicada, and we have a Massospora diceroprocta from Arizona, where we need to confirm whether or not they're the same. But it appears that some of them are genera specific. Most of them are genera specific, and some of them are kind of family specific. But it's clear that there was, this coevolutionary history dates back, to the earlier ancestors of these cicadas. So I would love to do a co-phylogenetic analysis once we get enough Massospora species to do that. But that's something I've been thinking about because we've done that with our ambrosia beetle fungi. It's always interesting to look for evidence of parallel cladogenesis.
Charles snuck in a comment and a question as well. The comment is fantastic and breathtakingly complex. The question is, how long are spores viable in the soil?
Well, spores are definitely viable for at least 17 years if infections are occurring in that vertical emergence chamber. Like, again, coming back to Matt's question, whether or not, are they infected on the way down? Do we find evidence of the latent infections? But we also know that they don't need that dormancy period before they germinate, because experimentally, people have taken spores from one brood and infected cicadas from another brood the next year. So they can infect, in 1 year's time. Interestingly enough, a lot of the annual cicadas we see in the East don't have Massospora infections. And I'm not sure why. Like, they don't even have their own Massospora species. The dog day cicadas, maybe it's too hot for the fungus to, maybe it was just a dead end for that fungus. So there's so much to answer in this, and hopefully NSF sees some value in that.
Thank you for sharing your current research and for all the information on your further research and opening the doors to questions and ideas on this fascinating behavior and this fascinating relationship. Are there any other questions before we say goodnight to Matt?
Well, I appreciate your time, and
I thought Matt answered all of his questions. Oh, it was. Okay, never mind. Sorry. This was great, by the way.
Thank you.
I'm sorry to interrupt, Stephanie. I thought Matt didn't answer all his questions.
No.
Sorry.
Taking care of the two Matts there. That's awesome. No, Kathy, I see you're back here on the call. So if you'd like to close out the meeting, I will mute
Oh, I'm
myself.
sorry, Steph, you do a great job. I'll let you finish. But sir, this was terrific. This
Thank
was
you.
great stuff.
All right. Well, I appreciate the invite and look forward to interacting with you all. Feel free to reach out to me on Twitter. Pretty active there, so it's a good way to connect.
Maybe, I'm sorry to interrupt, Steph, but maybe we'll get a refresher from you just before our brood so that we collect things in the way that's right for your project.
Okay, that sounds
Does
good.
that make sense? Because we
Absolutely.
really, we certainly can help here. Sorry, Steph.
No.
All right. Thank
Good
you.
night, everyone. Thank you for joining our meeting this evening.
Bye.